Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about the Meselson-Stahl experiment. And so after Watson and Crick came up with the shape of DNA, this characteristic double helix, the next thing to unlock was how does it make a copy of itself? And so there were immediately three proposals as to how it might copy itself. Number one, it could kind of unfold in the middle, and then we could copy new templates on either side. This is kind of what Watson and Crick proposed. There's another, what's called a conservative theory, that somehow the DNA was kind of wrapped around these histone proteins, and then it would make an exact duplicate of itself, almost like a photocopier. And then there's the dispersive model that maybe it would chunk itself into little 10, 12 nucleotide segments, and then each of those would be copied. And so what Meselson and Stahl did is they figured out which of these three is right. And in this picture, down here at the bottom is, it was later figured out that this is Martha Chase, who's also famous for that Hershey-Chase experiment. And so what did they do? Well, basically what they did is they grew E. coli, so this is Escherichia coli, or a bacteria, and they grew it in heavy nitrogen. What does that mean? Well, nitrogen normally has an atomic or a mass number, of 14. That means it has 7 protons, 7 neutrons. But heavy nitrogen is going to have an extra neutron, and so it's slightly heavier. And when you spin it in an ultracentrifuge, DNA that contains this heavy nitrogen is going to be moved to the bottom, and those that contain lighter nitrogen is going to stay up near the top. And so we can look at the density of the DNA to figure out what nitrogen's actually being added. And so if you think about it this way, thought experiment, let's say we started with heavy nitrogen, and then we were to copy that. So as the bacteria make copies of themselves, they're going to unzip that DNA in the middle, and since they're just in nitrogen 15, you're going to have 2 strands of DNA, but each of those are going to be all heavy nitrogen, so it'd go to the bottom of a centrifuge tube. If they were to copy themselves again, the E. coli, and make new template strands, 100% of them are going to be of the heavy nitrogen. And so what they did is they bred them in there until the point at which they were all nitrogen, all heavy nitrogen. They then switched them to E. coli living in the lighter normal nitrogen, and then they observed what happened with each generation. And so basically the lighter nitrogen in my little simulation will color it an orange color. And so bacteria on generation 0, we'll say, are going to be 100% heavy nitrogen, or 100% N15. As they unzip and make copies of themselves, that new template strand they're going to add is going to contain nitrogen 14. And so at this point, so kind of to keep track of everything, we had 100% heavy nitrogen the first time. On this first generation, now we're going to have 100% of the strands are a hybrid between the two. Half of them are heavy nitrogen and half of them are not. We're then going to go through another copying process. And so at this point, we've got one hybrid strand, another light strand, and then a hybrid strand. And so at this point, it would be 50%, if this semi-conservative theory is right, 50% light nitrogen and 50% of the hybrid. Now think in your head, if we were to do one more copy, which I don't have room for here on my simulation, well, this strand would create a hybrid, but the next 6 strands, as they split in half, are going to form light nitrogen, and this would form a hybrid. So now we'd have a 75% to 25%. And so that's what the semi-conservative would predict, and that's what they found in their experiment. So we had 100% heavy nitrogen in the first, 100% hybrid in the second, and then it split 50-50. Again, this is where my simulation stopped, but then it went to 75-25. And if we were to keep going, then it would eventually drop off to all nitrogen. And so what did that tell them? It told them how DNA makes a copy of itself. It simply splits it half in the middle, and then we're going to add new template strands on either, or we're going to read that template to create new strands on either side. And so that's the Meselson-Stahl experiment. It's very elegant. It's also very important, and I hope that's helpful.